The overall goal of the following experiment is to show an extraction methodology to obtain structural extracellular polymeric substances, or EPS, from biofilms, specifically from aerobic granular sludge.
This method can help answer key questions in the granular sludge field, such as understanding the granulation process and analyzing the EPS composition. The main advantage of this technique is that the presence of gel-forming polymers in EPS can be tested in a short time.
Centrifuge the granular sludge at 4000 times G and 4 degrees Celsius for 20 minutes and decant the supernatant. Collect the granules in the pellet for the extraction. Preheat 150 milliliters of tap water in a 1000 milliliter glass beaker on a magnetic stirrer to 80 degrees Celsius. Transfer 3 grams of granules in a 250 milliliter baffled flask and fill up the flask to 50 milliliters with demineralized water. Add 0.67 grams of sodium carbonate decahydrate into the flask to obtain a 0.5% weight per volume sodium carbonate concentration. Put the flask containing the mixture into the water bath. Cover the flask and the beaker glass separately with aluminum foil to prevent evaporation. Stir the mixture for 35 minutes at 400 rpm and 80 degrees Celsius. Transfer the mixture into a 50 milliliter centrifugation tube. Centrifuge the tube containing the